Well, thanks, Bengt, for giving me this opportunity to talk about our recent work. So it's a great opportunity. It's a marvelous conference, and happy birthday. I'd like to start, actually, by going back to what we were doing about 10 years ago, and this was a collaboration with Bengt and with Piero Baglioni, who was also in the audience. But before I do that, I'd like to just mention that what we do, we make chemically modified DNA and RNA. We use a chemistry, organic chemistry, to make modified DNA and RNA for various biological and nanotype applications. And we couldn't do that without the tremendous pioneering work that's been carried out by people like Bruce Merrifield and Marv Carruthers. They really are, they're the pioneers of the field. Now, that's important, but what's much more important is the contribution that these guys have made to biology, because modern biology depends so heavily on access to chemically modified DNA and RNA, as we all know. So, this is a fantastic example of how solid phase synthesis, the details here don't matter, the chemical details don't matter, how this can contribute to all different areas of science. So it was about almost 10 years ago now that we were working on a project with Bengt and with Piero looking at assembling DNA nanoconstructs. And we came up with a methodology whereby we could attach DNA strands to a central node, making Y-shaped pieces of DNA with specific sequences, and we could take a whole variety of these and mix them together, and there would be a bottom-up assembly into specific nanoconstructs that would occur to give very tightly packed, if you like, hexagonal nanoconstructs composed entirely of DNA. We were able to get this to work, and an individual piece of this would look like a kind of a hexagon with various arms, and the arms would link together to build nanoconstructs. This is the same object if we rotate it by 90 degrees. The size are 1 turn of bDNA, so they're the smallest flat piece of DNA that you can construct, a single turn, 10.5 base pairs or whatever. And we were able to assemble them uniquely into various structures, and for example, this one we call DNA naphthalene, using 10 of these nodes, and this one we call DNA anthracene. So you can see how you can program assembly of nanoconstructs, again, bottom up. Of course, we have to show that we've got these structures, and AFM was very useful in this context. You can see here these 2 6-membered rings together to form this DNA naphthalene, and this one we call DNA benzene, and thanks to Piero for these AFM images. Importantly, this is a scaffold. Because it's a scaffold, we can attach things to it, we can attach molecules to it, and we can encourage these molecules to talk to each other. A simple example of this is we have 2 fluorophores, a fluorophore here and one here. We put them within fret distance, and we can do fret between the fluorophores, and we can show that they are in this kind of relative orientation, so they talk to each other. In order to do that, we have to address double-stranded DNA. And the work that we did around 10 years ago, and there were many other people who worked in the same area, was to find a way of addressing double-stranded DNA. And the way that we did this was to construct a triple helix, whereby the duplex had in its major groove a 3rd strand. The important thing here is that the 3rd strand has sequence-specific recognition of the duplex. So we have a modified monomer that can recognize GC, a different one for AT, TA, and CG. So we can read the DNA sequence in the major groove using a 3rd strand. So we can then address a specific part of this structure specifically. Now, this is not perfect recognition. It's not as good as Watson-Crick base pairing. I don't have time to go into the details, but it does work. But there was always a problem assembling these kinds of nanoconstructs. You can imagine you're trying to assemble a nanoconstruct from DNA which is this shape, but the 2 ends don't always come together to form a duplex, and you can trap non-productive constructs, which is not what we want to do. The non-productive constructs are shown here. The one that we want is shown here. So the idea is, can we fold these things into the correct shape and then keep them in that shape and use them as building blocks for bigger nanoconstructs? So one way to do this would be to chemically fix these things. So you would make this mixture. The best we could do was about 82% of the correct one. The idea now is, can we actually take that, can we cross-link it chemically, and then purify it by fairly harsh methods to get rid of all the incorrect sequences? So we needed, in order to make our building blocks bigger and to get pure building blocks in this type of approach, what we needed to do was to find a chemistry that we can use to do this. Now, of course, these are big molecules. These are DNA molecules, and so you have to have a very selective chemistry. And for this, we turned to, well, we tried a number of things, but the one that we arrived at eventually was the reaction between a terminal alkyne and an azide to form a triazole catalyzed by copper-1. And for this, again, we have to thank the pioneering work of Barry Sharpless, M.G. Finn, Valery Fokin, and Morten Meldal for developing this type of chemistry to enable us to do this. So we applied this chemistry to DNA, and we used it to link together DNA strands to fix them. Now, it's really good. This chemistry is great for this kind of application for a number of reasons. First of all, azides and alkynes can be attached conveniently to nucleic acids. They don't react effectively with other functional groups, with one or two exceptions. So they give selective chemistry. The reaction goes extremely well in water. It goes really well in water. So this is obviously crucial for DNA. And you can switch the reaction on by adding the catalyst once you've assembled the correct structure by slow annealing. So the reaction only occurs when you want it to occur. And the product is a stable product. So we have a chemistry that we can use in this context. And this slide will show us how we did this. So we took, as an exemplar, we took 6 different oligonucleotides. They had alkynes and azides at the end, matching functional groups at the ends. And we add copper, and we perform 6 simultaneous click reactions on this construct to link everything together. Now, I'm not going to show you detailed chemistry in this presentation because there's no time, and I want to show you the applications rather than the mechanics of it. But just for those who are interested, these are the kinds of functionalities that we used on nucleosides to build these nanoconstructs. The point is that when we did that, we could take our crude mixture and we could now separate our desired structure from all the non-productive structures by denaturing gel electrophoresis and obtain the correct construct in a pure state. So our bottom-up approach can be used now. We can build bigger and bigger building blocks to assemble DNA, nanoconstructs. We performed an experiment where we took a long piece of DNA, a 7-timer piece of DNA, with an azide at one end and an alkyne at the other. We add copper, and this 7-timer DNA cyclizes to form a cyclic single strand. We then take the complementary 7-timer, alkyne and azide at the ends, and we use this one as a template, and we template the addition of the second one to form a DNA catenane, add copper, and we fix it. So by doing that, we can make quite large cyclic DNA nanoconstructs which have got a triazole linkage in this position and one in this position. The yellow pieces here are mismatch base pairs. You need those mismatch base pairs to allow this 7-timer DNA to bend into a circle. They're effectively hinges. So we published this in 2007. And then we went on to the other extreme of size. We took a series of hairpin loop oligonucleotides where we have a hexaethylene glycol linkers at one end, an open loop, complementary base pairs, alkyne and azide at the other end, and we add copper to cyclize. One thing to notice is the chemistry is very, very efficient. So when we do this, we take the hairpin, we add copper, we get the product, a 5-mer product, a 4-mer product, and a 3-mer product. So we're making smaller and smaller pieces of cyclic DNA using this click approach. And we went further than that. We then went to make even smaller pieces, 2 base pairs and 1 base pair. And I'll show you the significance of this in the next slide. We were able to show by this approach that if we cyclized the hairpin loop to make a cyclic construct with a triazole long linker at one end and a hexaethylene glycol linker at the other end, we could observe within this structure very, very stable base pairing. In fact, we formed a duplex, which we could follow by a technique called UV melting, which was very stable and couldn't be melted in aqueous buffer below about 75 degrees centigrade, even though it's only 2 base pairs. In fact, we had to add formamide to melt this little duplex, which is very unusual for DNA. And we were able to show that this duplex has a normal bDNA-like spectrum, CD spectrum, so it has a helical shape, even though it's only 2 base pairs. We were able to show that it binds to various drug molecules which bind to bDNA. So it's a cyclic mini duplex. It's the smallest one that you can really get and have normal DNA-like properties. Because when we go further than that, just to show that the duplex looks like this, here we have base stacking between the base pairs, hydrogen bonding, and the two linkers. When we did the same experiment with a single base pair, we did not observe any base pairing. And the reason for that, and I think people who work in the nucleic acid field will see this straight away, the reason is that there's no base stacking. If you only have one base pair, there's no base stacking, so you completely lose the contribution of base stacking to the stability of double-stranded DNA. So we could not observe, this is the smallest duplex that we can observe using this methodology. This is an interesting thing to do, to make these nanoconstructs and study them, and there is scope for studying unusual base pairing and modified bases in this kind of way. But we wanted to see if we could apply our methodology to something more biologically relevant. DNA ligase is an enzyme that links together DNA strands to form double strands and stitches together the backbones. It's a very important enzyme in biology. It's used in replication and in DNA repair. And we were interested to find out whether we could develop a triazole-type methodology to do a chemical linkage between DNA strands in order to mimic a ligase enzyme. So the idea was that we could take pieces of DNA and we could link them together chemically using this click reaction, using a splint oligonucleotide to hold them together to make sure that they link in the right order, and use this instead of using an enzyme. Now, one reason that we wanted to consider this approach was that it's been known now for just only 4 or 5 years that there are a number of modifications to DNA which control gene expression by an epigenetic mechanism. And they involve modified bases, hydroxymethyl C, formyl, and carboxy C. We've known about methyl C for a very long time, but these other bases have recent entrance into the party. So we thought, it would be really interesting to develop methodologies, not just to incorporate these kinds of modifications, but to incorporate all different kinds of chemical modifications into big DNA strands for various biological applications. And you cannot do this by conventional PCR, because PCR only works with normal triphosphates, A, G, C, and T, so it erases this extra level of information that you would incorporate using a PCR reaction. So the idea was, can we develop a linkage between two sugars that would mimic a phosphodiester group and would enable us to link together DNA strands? So we started with something that we could make very quickly in the lab, and this doesn't look anything like a phosphate group, but it is a linker between two sugars. We basically, if you compare it to normal DNA, you can see that it's longer, it doesn't have the negative charge, and it has a rigid amide bond. But anyway, we made that, we modeled that structure, and you can see that the triazole linkage is here. This is normal DNA, this is the triazole DNA, this is just a rotation of 90 degrees, but it should be able to allow base stacking and base pairing. So we took two pieces of DNA, one with an alkyne at one end, one with an azide at one end, and we clicked them together. And we carried out PCR to amplify the DNA. So the question was, can DNA polymerase enzymes read through the linkage? And when they've read through the linkage, do they produce the correct sequence, the same sequence as the original strand? So, first of all, we were very pleased to see that the chemistry to make the DNA was extremely efficient. This is the template strand, the splint. These are the reactants, and this is the crude reaction mixture to give a clean product. When we took this piece of DNA, which was 115 in length, we used 2 PCR primers and we amplified it, and we proved that the DNA polymerase enzymes can read through this unusual linkage and produce a PCR product fairly efficiently. But that doesn't give us the whole story. We then had to sequence the PCR product to see if the copying had been correct. And we had to sequence a very large number of clones to satisfy the editors of JAX, which we did 130 clones. Every time we got the same answer. The answer is at the site of interest, we got this sequence. Well, that doesn't mean much until we look at the next slide. In the next slide, we can see that what we'd actually done is we made the linkage, we carried out PCR amplification, and instead of having the 2 T bases, T and T, at the site where the linkage was, we only had 1 T. So the polymerase enzyme had looked at this linkage, And what it had done is it had jumped over one of the two thymine bases at the click linkage. Now, you could argue this doesn't matter because you can always model into your design the fact that you can eliminate a base during copying. So you can use this in a practical sense. But in reality, we wanted to develop a linkage which was actually biocompatible, that wouldn't do this. It would actually behave completely normally. So we went back to the drawing board. We redesigned our system. This time we incorporated an oxygen atom here, like normal DNA, a CH2 group here, and we made this linkage. We had to bear in mind that you have to make a linkage which is easy to make, so we are restricted in a sense of what we can make here. And we carried out the same kinds of experiments. And again, this is a chemistry slide, and I'm not going to go into detail on this. This just shows people who understand nucleic acid chemistry that making this linkage is very easy because you can put an azide at one end of a DNA strand and an alkyne at the other in a very convenient way. So we did the same experiment as I've described previously. We did the chemical linkage, we did the PCR amplification, which worked beautifully, and we did the sequencing. And this time it works. This time we get perfect read-through of the triazole linkage. So this very unusual linkage, which looks nothing like a phosphate group, is treated pretty much normally by a whole variety of different normal and thermostable DNA polymerases. Okay, so next we push this a little bit further. We take a linear 100-mer oligonucleotide, azide and alkyne. As you can see, we can get this very pure by HPLC. We cyclize it to make a triazole, we cyclize it to make a triazole linkage, and we use an enzyme that can continuously read through the linkage, going round and round and round. Rolling circle amplification, it has to pass through the triazole many, many times. And we compare the amount of product that we get from this RCA reaction with normal DNA, and they're pretty much indistinguishable. So that's a good test of the robustness of this linkage. A little bit more of a stringent application, we take a template DNA, a primer, and we read through just once. So now rather than letting PCR rescue anything that's not quite right, we do a stringent thing. We try to read through a linkage once, and we work out how long it takes to read through the linkage once. And what we find when we do that is that if you wait for 2 minutes, you can read through a triazole linkage just the same as a native linkage. But if you take a snapshot at a much earlier time scale, you can catch the situation when the triazole DNA has not yet been read through. So the polymerase is slower to read through the triazole linkage than the normal linkage, but 2 minutes is not a long time, and 2 minutes gives you the correct product. Not only that, but we put the triazole linkage in a DNA construct, and we transcribe from it. And we found that T7 RNA polymerase can read through the triazole linkage and can make RNA. So DNA polymerase can read through the linkage, and RNA polymerase can read through the linkage. Other results, which I'm not showing today, demonstrate that reverse transcriptase, RNA to DNA enzyme, can also read through the linkage. So all three, can read through the linkage. We confirm all our results by mass spectrometry, in addition to sequencing. But what about bacteria? So we take a plasmid and we put a triazole linkage in each strand of the antibiotic resistance gene that's required for growth of E. coli, and we transfect the E. coli. So if the E. coli survives, it means that the E. coli is happy to treat this triazole linkage as normal. And indeed, that's the case. This is in collaboration with Ali Tavasoli in Southampton. Basically, we get 96% of the colonies that we would get from normal DNA when we use our triazole-linked plasmid. We repeat the same experiment with a strain of E. coli which is deficient in DNA repair to see if we're repairing the scar or we're just normally reading through the linkage. And that works. So this experiment tells us it's unlikely that DNA repair is chopping out the mistake and putting something correct in there. The enzymes in E. coli are probably just treating the triazole as normal, which is entirely consistent with previous results. In order to get around any issues about selection pressure, we repeat the same experiment whereby the plasmid carries a gene for a fluorescent protein. So now that the E. coli doesn't need to express this gene to survive, but if it does express the gene, the colonies will be fluorescent. And indeed, it works fine. the colonies are fluorescent, the triazole colonies and the native colonies look very much the same, so the gene is being read through in E. coli. And we take the plasmids back out and we sequence them and we demonstrate that we're getting the correct sequence from the plasmids. So everything's fine. And the final step in this study, which was just published quite recently, is to do the same thing in human cells. So we transfect human cells, we put the mCherry gene inside human cells, and what happens is that we observe that the human cells become fluorescent. So eukaryotic cells are also able to recognize these linkages as being effectively normal. So what's the structural basis of this? Well, here we have in orange, we have a normal bDNA duplex, which we determined the structure of by NMR in collaboration with André Dolman. And in blue, we have the same sequence with a triazole linkage in the center. So we're comparing the structures. And the first thing to notice is that they look identical except at the site of the triazole linkage. So normal bDNA, no problem. But at the site of the triazole linkage, we have a little bit of a discontinuity. We have a triazole protruding in between the major and minor groove. But very interestingly, the position of this triazole nitrogen here is identical almost to the position of a phosphate oxygen, potential hydrogen bond acceptor. So one point to mention, actually I'll just go on to the next slide and show this in a little bit more detail. So what you can see here is that the triazole here is in the middle of the duplex, and as it comes round and we expand the picture, what you will observe is that the triazole nitrogen is really, really close to a phosphate oxygen of normal DNA. So what? So what does this tell us? Or what does this suggest? What it suggests is that when you look at a DNA polymerase, it binds to a template DNA strand. Our template has got one triazole in it. The interactions between the template and the DNA are mostly hydrogen bonding, one or two electrostatic interactions, but mostly hydrogen bonding. The point here being that triazoles are not charged, but phosphates are charged, but doesn't really matter. But the key point is that if we probably don't even lose a hydrogen bond because the triazole nitrogen can compensate for the phosphate oxygen. We did a very recent experiment, and this is really, actually, this is not proven yet, but it's very suggestive, where we methylated that triazole nitrogen. And when we methylate that triazole nitrogen, what we find is that the read-through is really slow. So just a simple methyl group blocking this potential for hydrogen bonding is deleterious. But there is a problem with the triazole linkage, and the problem is as follows, that the DNA duplex becomes less stable at the point of the triazole. The base pairs around the triazole start to fray more than normal. It doesn't seem to affect its biological properties, but we wanted to see if we could fix that problem. We had antisense therapeutic applications in the back of our minds when we did this. Can we restore the stability of normal DNA that we lose from the triazole in a different way? So normal DNA is more stable than triazole DNA when we compare them directly. So in order to do that, we do nuclear-based compensation. We take our normal cytosine base and we add to it an extra hydrogen bond above in the major groove. So we have 4 hydrogen bonds, and the idea is that the 4th hydrogen bond will bind the bases together and compensate for the instability caused by the conformational flexibility at the triazole. And indeed, it works beautifully. When we do that, we find that our what we call G-clamp triazole DNA is more stable than standard DNA. So we restore the stability. We also have shown that if we carry out our standard experiment, whereby we make the linkage, read by PCR, and sequence, although I'm not showing you all the details, this G-clamp triazole, this doubly modified DNA linkage, behaves as normal. We can read through with polymerases, and it gives the correct sequence. We next turn to RNA. RNA is more challenging to synthesize than DNA because the very properties of RNA that make it such an interesting and important biomolecule make it more difficult to synthesize. The 2' oxygen interferes with the 3' phosphate group. We get chemical reactions that can cause migration of the phosphate and cleavage of the backbone. And if we're not careful with our protecting group strategy, we can have problems. Actually, protecting groups that you have to use here are bulky, so they interfere with the chemistry. So RNA is more challenging to make than DNA. So in this context, we decided to make a hairpin ribozyme. we took 3 pieces of RNA, around 35 in length, which we can get very pure. And we have alkynes and azides on them, so that when we assemble them into a ribozyme, the stem loops of the ribozyme enable a click reaction to occur between them to make a chemically linked construct, which is a 100-mer ribozyme, and then we use that ribozyme to cleave a substrate. Making a 100-mer RNA in a linear solid phase manner is difficult. So this is the crude reaction mixture here. So we take our 3 pieces of RNA and we assemble them against each other, and this is the crude reaction mixture, and this is the 100-mer RNA. It's a very clean reaction. We can analyze by mass spec and gel electrophoresis to prove we have the right thing, but of course the functional test is the important one. We take a substrate, we add the ribozyme, and it cleaves rapidly to give us the cleaved substrate. We did the same thing with a hairpin ribozyme, with a hammerhead ribozyme, which is much smaller, just to show that if we put the triazole linkage in the hammerhead ribozyme, fairly close to the sessile bond in the substrate, it actually maintains activity. So the click linkage that we put into the RNA, into the ribozyme, does not prevent the ribozyme from cleaving its substrate, depending on, and this is quite close to the active site. Now, we were wondering, is this a general property? Can you just put anything between 2 sugars and it will be read through by polymerases and everything will be fine? So we took a 3rd generation where we have a shorter linkage, in a sense closer to the length that we have in a normal linkage. And we did a similar study. So we synthesized our DNA with this linkage by taking an azide and an alkyne DNA, clicking them together, very efficient reaction again. Chemistry always works beautifully in this context. PCR amplification works. So far so good. But this new triazole is no better in terms of duplex stability than the previous one. Even though it's shorter and similar in length to a normal linkage, it still gives a less stable duplex. And this is not yet published, but the point I want to make now is that when you actually amplify that piece of DNA and you sequence the DNA, we get all sorts of mutations around the site of the triazole. So that this linkage is not fully biocompatible. So it's not just a case of just making any triazole between 2 sugars or any linkage and it will work. It's more subtle than that. So we were lucky with the one that we designed. We were lucky. There's no question about that. Now, the copper-catalyzed reaction is a fantastic reaction. But in certain contexts, in biology, for example, it's sometimes difficult. Say, for example, if you want to ligate pieces of DNA in cells, copper is a problem. It's difficult to maintain integrity of biological systems in the presence of copper. Problem is that the reaction without copper, the Huisgen reaction, the original reaction, is exceedingly slow. So if we take this hairpin DNA with an alkyne and azide and we incubate it at various temperatures in the presence of aqueous salt, we can make the reaction work, but the reaction takes a couple of days at 80 degrees centigrade, which is not an option for biological applications. Now, this kind of problem has been addressed very nicely by Caroline Bertozzi in a non-DNA context, in a biological context, where she's carried out click chemistry in a cellular context on glycans. And the way that she did this was to use activated triple bonds, strained alkynes. So we tried this on DNA. So we take an azide DNA and an alkyne DNA, and we join them together. Now, no catalyst is required. We can't switch the reaction on. It's always on. But when we template the reaction, we increase the concentrations by templating. The reaction occurs, and we can have a strain-promoted click linkage between 2 DNA strands. If you want to look at this on a gel, here we take the reaction I've just described and we put the reaction mixture on the gel, and as fast as we can put the reaction on the gel, the reaction has gone 100%. Now, we can slow the reaction down by deactivating the cycloalkyne, and we can make it a slow reaction, or we can actually introduce mismatches into the template. And if we do that, then the hybridization doesn't occur properly and the reaction doesn't work. So the reaction is sensitive to mismatches, so you can envisage it in context of mutation detection, it could be useful. But these reactions are really fast and efficient. we did a lot of different cross-linking reactions between DNA strands. And one example I'm showing here is where we put the cyclooctane in between two sugars in the minor groove to link together two DNA strands. And we end up with a very stable duplex, permanently linked, with a minor groove cyclooctane. But we did lots of examples. And we did lots of other click reactions. We used Diels-Alder chemistry, tetrazine type chemistry, and we broadened this to a number of different systems. When we use a cyclononine, which is a stereochemically cleaner version, we can see again the reaction is gone within a minute to 100%. And we can do the reaction on solid phase. Solid phase has got many advantages over doing a reaction in solution. You can force a reaction to completion by using large excesses of the solution phase oligonucleotide. So we can do the reaction on solid phase, and we can get very efficient solid phase assembly of quite big pieces of DNA on a solid phase. And then we can cleave it off and utilize the DNA. Coming close to the end of the presentation now, but I wanted to just mention a couple of applications that we've looked at or are still looking at, and this is very much work in progress. This is all very recent work. One application is that we took a gold nanoparticle with a single azide DNA on it and another gold nanoparticle with a single alkyne DNA on it, and we held them together using a complementary oligonucleotide splint. Now, the way that we synthesized these gold nanoparticle DNA conjugates was to add a single DNA strand to each nanoparticle, using a very highly dilute DNA and using gel electrophoresis to separate the nanoparticle with a single DNA from nanoparticles with lots of different DNA strands around them. So you could say that this nanoparticle is a monovalent gold nanoparticle where the chemical bond is coming from the DNA strands that we're holding together. So when we catalyze that reaction, we produce two gold nanoparticles held together with a single piece of DNA, which is permanently linked by a chemical bond. So we can carry out TEM studies, and we can observe the separation between the two gold nanoparticles, which is consistent with the length of the oligonucleotide between them. So it's a valency. This is, if you like, a monovalent gold nanoparticle. And we've done the same thing using a gold nanoparticle with two DNA strands, which is divalent. And we can create two single bonds, one gold nanoparticle with two bonds to join three gold nanoparticles together. I just didn't put the images on this overhead. So we can control this stoichiometry of linkage. What we can also do with the copper-free chemistry, going back to the beginning of my presentation, is that we can take a DNA nanoconstruct, this one here, which is a 7-timer duplex, which is rather like the ones I showed at the beginning of my presentation, and we can put an arm on it, and the arm can have a complementary oligo on another specific DNA nanoconstruct, and we can join them together, and then we can click them together using this copper-free chemistry. This doesn't, by the way, this didn't work using the CuAAC reaction and monovalent copper. We could only do this with the copper-free chemistry. And we can join together either 2 or 3 or strings of these DNA nanoconstructs using this kind of chemistry. So we can assemble in a controlled way DNA nanoconstructs. The very final thing that I want to talk about is, I think we all know that it's Benkt's 70th birthday. But I don't think many people in the audience know that he has a new baby. He has a new baby, which is a new piece of physical chemistry that we've been developing with him. And it's to do with stretching DNA. So if we take DNA strands and we have on each strand a microparticle, we can use optical tweezers to pull the two strands. This is well-known technology. And we can pull them apart, and we can measure the forces that are required to pull the DNA strands apart. And we can work out from that the properties of the DNA that we're studying. One of the drawbacks here is that when we pull the DNA strands apart, we also pull apart the particles. And then we have to find another pair of particles, and this is Niklas's work. Niklas has then got to go and find another pair of DNA strands that are held together, and they're hard to find, and repeat the experiment. By very simply clicking together the ends of the two DNA strands, as I've shown here, using click chemistry, we can hold the two particles together, and we can repeatedly stretch, relax, and we can observe quite subtle changes in the forces required to pull these strands apart. One reason that we find that this is interesting, it enables us to observe stretching forces which would otherwise be hidden. And we've published with Bengt 3 papers recently in this area to show that we can isolate a stretched form of DNA which still has base pairing. I'm not going to describe this any further because it's really Bengt's, as I said, it's Bengt's new baby, and that's for him to do. But this is a really nice application of click chemistry. Finally, I'd like to thank you very much for your attention, and just to also thank the people in my group, my new group in Oxford, who've contributed to this project. The person who's done the majority of the work is here, Afaf, and these are the other contributors listed on the overhead. And very importantly, BBSRC, the UK grant funding body, has been very generous in giving us quite a large amount of money to pursue this particular area, and we're about halfway through a 5-year project funded by BBSRC. ATD Bio is a small company that's helped to sponsor the work. And for those of you who are interested in undergraduate teaching, and I'm sure most of us are, we've constructed on the internet a nucleic acid textbook, which covers all different aspects of nucleic acid chemistry and biology, and it's free to access for anybody who wants to use it in any of the teaching courses. Thank you.
Thank you, Tom. You are on time, so I will allow a couple of questions this time.
Thank you.
It was a fantastic presentation, I think, on intelligent design. What do you say, Bengt? You have a question here. I think it works.
Okay. Does it? No? You have to signal to the machine room.
His hand's up now.
Okay. I'm thinking of Tom, when you... Beautiful talk, by the way. I realized that it was well worth inviting you.
Thank you. That's a relief.
When you incorporated the triazole into the backbone, despite the fact that you had the right slack, the right length, you get all kinds of mutations. Have you thought about the why?
Well, there are a number of possible origins for that. One of them is that, particularly with the linkage that we showed, which had an amide bond, obviously there's very restricted rotation about that amide bond, and it wasn't the cleverest first design, but it was something we could do quickly. And obviously, the chances are that it's difficult for the DNA to adopt the correct conformation within the active site of the DNA polymerase to allow read-through. A second possibility is that you get fraying because the stability of the duplex is a little bit reduced at that point. So another possibility, and maybe a second contributory factor, is that the DNA polymerase is not able to grab onto the next base because it's just not fixed properly, you know, against the complementary triphosphate.
In view of Roger Kornberg's talk, I was thinking, could it be that you don't get the proper base-base stacking?
Well, yeah, I mean, one thing that we have not yet done, and we're actively pursuing, and it's a really obvious thing, is a crystal structure, an X-ray structure. The NMR structure demonstrates that the orientations of the bases are about right, but clearly there is some fraying, so there's some instability. But it will be really interesting, because NMR is a fantastic technique, but complementary to that, to see a crystal structure and see what's actually happening at the site. And we're actively pursuing that. We haven't yet got good crystals, but we're pursuing a number of options. Hopefully, when we come back to your 75th birthday party, we'll have an X-ray structure. Yeah, yeah. If I'm invited to that, of course. Yeah. Yeah.
We take one more question up here. So, just a second, and you will have the microphone.
They're always great when I hear your talks. Is it working?
It's on.
There's a lag in Sweden? No. Oh, okay. I understand. I followed pretty closely, and I also read that paper you and Bengt wrote in PNAS. And just about fell off my bed where it was so amazing to me. So you stopped short of that. Somebody asked maybe. And anybody who likes DNA likes evolution, and so maybe Bengt, can you just get up there and could Bengt tell us what the conclusion was in 2 seconds? No. It's too hard. Okay, but read that paper.
Yeah. Okay. Thanks, Barry. Thank you.
It's an excellent topic for the panel discussion this afternoon.
Yeah.
Thank you.
Yeah.
So thank you again, Tom, for a
Thank you.
fantastic presentation.